So basically wanted to talk about the sexy thing of the moment, which is artificial intelligence, which is machine learning and computer intensive methods in randomized controlled trials. And so what is artificial intelligence? What do we understand this is? We'll talk about it, but I will talk about how we use artificial intelligence methods and how we use computer intensive methods. And there's a difference between computer intensive methods and artificial intelligence methods. And I'll define those. It's being used in drug discovery, in optimizing patient selection, precision medicine, risk-based monitoring, and in secondary analyses and doing secondary analysis. The primary analysis is not touched with this. Primary analysis is always that crude analysis, t-test, Fisher's exact, log rank test, whatever. So these artificial intelligence methods are used for some of the other aspects of doing randomized controlled trials. Computer intensive methods are used in secondary modeling, secondary analysis using genomic information, multiple imputation to handle incomplete data, as Jay Seelan mentioned, and also in optimizing randomization. We'll talk about some of these computer intensive methods. So what do we understand by, I don't know why it's not. That's not. Let's pair again. Okay. So what is artificial intelligence? So first of all, we have to understand that because many people throw out these terms and are not talking about the same thing. So it's simulating human intelligence. That's why it's artificial. Using machines that are programmed to think like humans as best as possible and mimic human action. Okay. So there are technologies that enable computers to perform tasks that require intelligence, like decision learning. So learning is one human intelligence process, problem solving, and decision making. Okay, and so they're trying to mimic these sorts of tasks or actions that require some thought. It relies heavily on data to learn patterns, improve whatever the task is based on its learning. Okay, so these algorithms that machine has learned enable these systems to analyze vast amounts of data faster, more efficiently than humans can do, and make predictions or take actions based on those patterns. There are all sorts of artificial intelligence things, but I'm only going to talk about the ones that are pertinent to randomized controlled trials. I'm not going to talk about robotics or chatbots or Alexa or Google this or whatever. That's something else. Or generative AI and writing yeah, I guess you could do it, writing protocols using AI, etc. But AI is transforming trials by enhancing separate aspects of trials, from design, patient selection, patient allocation, so sometimes using for secondary data analysis and outcome assessment. AI algorithms are used mainly to improve efficiency because they can do things faster, more efficiently sometimes than humans can. Ensure study integrity, maybe reduce costs, potentially. So there's an initial investment that is expensive, but then if you use it a lot, then it's reducing costs, and then potentially accelerate development of new therapies. So in RCTs, we use them for multiple things. Image recognition. So for example, if I have related to the outcome, I'm looking at strokes and I'm doing MRIs, and to see if you've had a stroke, especially lacunar strokes that are not easily found. What do we have to do? We have to have multiple cross sections. And who is looking at it? Some human to figure it out. It's a heck of a lot more efficient to have the machine learn how to look for it and detect it, and to see changes in the area or volume of a lacunar area there. Much more efficient. Radiologists are getting upset because their jobs may be going away with this. So image recognition. Also, people may be using it to, when you're counseling a patient that is participating in your trial, and your trial is now on behavior change, and you're trying to get them to exercise or change a diet or this and that, and they have to come in to clinic, and the person that is doing the care and follow-up is interviewing and asking questions. People that are not following things tend to lie to you that they're following things. And what did we used to do in the past? We had lie detector things that somehow would measure how sweat you have, your vibrations or whatever. I don't know how these work. And they would say if you're lying or not. Nowadays, what do? They already have the consent that all interactions with the staff are going to be filmed. They film this. Now they have AI look at your movement on your cheeks, how you're, they're asking you this and you go like this. Now they're using that to maybe guess if you're following the intervention or not. So these are the types of ways that they're using image recognition to do this kind. So for diagnosing the outcome occurrence, for looking at adherence to the protocol, that's the kind of stuff that they're doing. So because that recognition of patterns and unusual patterns is what machines can do a heck of a lot better and objectively, supposedly, than humans trying to do it. Because that nurse or person that is doing the interview, how do you train that person into doing it? You can train a machine much better than you can train a person to be reproducible and reliable and objective in that recognition. So speech recognition also is, they would, they use this for converting spoken words into text. Where do we get that? Because when in some of the trials, we have events that are not easily detected. They are, the condition of the patient as per the physician medical records. And most of the medical records now are, dictated, or they may be also going to a person's, if the person has died and you want to do a verbal autopsy with the next of kin, you tape it, you film it, and now you want to take that and understand it, digest it. And so now that digestion is easily, more efficiently done with machines. But also to forecast future outcomes. And we'll see a little bit more detail on that. Who is likely to benefit of the intervention? We're using AI to, I say we, royal we, I'm not using it yet, to predict who is going to benefit or not. And identifying patterns and insights in large data sets, that we are doing, and I'll give you some examples of that. So when we talk about machine learning AI, now we're talking about different types of methods that are algorithms that are programmed into these machines, and they have different types of ways of acting. So we have what's called supervised learning or unsupervised learning or deep learning. Okay. So supervised learning is you know what has happened, and you train the machine to figure out ways that will give you that thing to happen. And so the desired output is known. So the person died or the person did not die. So what things would be predictors of that person dying or not? So you know that. But it's also used for image classification or spam filtering. In RCTs, we use it for adjudication. So we know the person is dead, or the person may have had a coronary heart failure, coronary heart disease, or sorry, hospitalization for heart failure. Yes, we do that. But they may have died suddenly, and we don't know if it was due to an MI or not. And so we have a bunch of records, medical records. We have the echocardiogram, we have this, we have that. And now somebody in an expert panel of adjudicators, cardiologists look at it and decide yes or no. Well, we could program the machine to have the protocol for classifying somebody as that, but there's so many different patterns. Yes, you can get those expert cardiologists, but the machine can always give you the same result like that, because we train cardiologists in a particular study. We have differences of opinion between two cardiologists on the same case, but they follow in the protocol. We can get the machine to do it more efficiently. Unsupervised learning is when we use it, it's finding patterns in unlabeled data. And we actually have used this to detect fraudulent data. And so we have all the records. So we have studies that have 4,000 people that have huge CRFs that are collected over time. So maybe 10 times 400 questions on 4,000 people. And how do we know if there's any data that's fraudulent? Can you have somebody human go through each and every one of these things? It would take years to do something like that. We have algorithms that we have worked on with some of these AI experts to detect fraud, and I'll give you an example of that. There is also the possibility of deep learning, and this is what's called artificial neural networks. In our brains, we have that networks between within our brains that make connections. And so when we hear something gets triggered and we make that connection, etc. These things are emulating our brain operation. They are artificial networks, and it's to use to analyze data. It's more for, speech recognition, image recognition, natural language processing, but it's used also as a black box prediction model. You throw in all of these variables into this model, you put the genetic code of this person, you put their behavior, you put all sorts of things, and they'll tell you whatever you're trying to model, like the likelihood that this person will be a success, or the likelihood that this person will have a heart attack, or the likelihood that this will happen. And so there are these models. They don't tell you, 4 times this plus that will give you this risk. It just says yes or no. Yes, or the probability that you will have it, or probability that you won't have it. So these are black box prediction models. And I'll give some examples of these. So it's used in multiple aspects of clinical trials. One of the aspects that pharma is very much using is in drug discovery and development. So basically trying to narrow the gap between the understanding of the disease and potential therapeutic agents. So there's many things that are in nature, many things that are chemical compounds, and they can train algorithms to decide, will this compound mix with this maybe work or not, because it changes this particular thing and changes that particular thing. So because it can analyze extensive data sets and look at patterns that we may not find as humans, it may identify potential new therapies and therefore enhance the predictions and efficiency. So pharma is trying to not have to do a bunch of preclinical work to figure out if something may work. And they're trying to become more efficient. And this is a paper that just recently came out in fundamental research on actually this. It's revolutionizing drug discovery. I'm not involved in this. I don't have any expertise in this. But what they say is, we can take the clinical sequencing data, the experimental text, molecular structures, we feed it into these models, and for them, features is variables. And they look at relationship with variables. They use these neural networks, and they come up with predictions that then will help them maybe diagnose disease or help them with whatever they call it, target acquisition or computational screening, and enhances this drug discovery process. So this is one place where it's being done prior to a clinical trial. This is to identify a potential new drug that may work using all the vast amount of clinical sequencing data, experimental text, etc., as well as chemistry. Now, we've talked already, they're using AI for select and optimal interventions. That's exactly what the smart adaptive design is doing. And that is precision medicine. So it's identifying optimal doses. So then in those smart designs, which one is the next one that I should give to this person, given that they've had this before? So now through computer intensive simulations, you could do it. But now we could also incorporate characteristics of the participant, and I could get characteristics of other people who have benefit from it. So now they're using AI prediction models to decide which is the next, not only dosage, but which is the next chemotherapy or the next medication that we should try on this person, because I can calculate the probability that it's likely to work on this person. So this is in early phase trials that they're doing that. So there's another place where they're using artificial intelligence. And these are in early phase trials. Okay. This is actually one place that many people find it very attractive. We have inclusion and exclusion criteria. And so how do I decide on that? Based on my experience, based on the literature, I said, well, and I don't want to have people that have renal disease, even though I'm studying HIV. I don't want to have people that have diabetes when I'm studying HIV because they may be potential confounders. So now what am I doing? I am homogenizing who I'm studying. I'm reducing the generalizability of my results because I'm studying a very narrow group of people. It's a very artificial group of people because the general population of people with HIV, also some of them have renal disease, also some of them have diabetes, also everybody's complicated. But if I include all of that, I make it so heterogeneous, it's difficult. What people do is, can I use artificial intelligence to decide what should be my optimal inclusion and exclusion criteria? So we want to balance who is likely to benefit from the intervention versus I want to be as broad and generalizable of my study results, so their external validity. How do I do that? From information from early phase studies, and I have to have individual patient data from other people in other studies that have done this. So it's hard to do unless you have access to that. Our institution has done over, in the last 25 years, has done over 60, 70 trials. And we have that data. That is our gold mine that we can use now to see. I can find from all of these, we would have over close to 600,000 people that have participated in some of our trials. So I now have data from 600,000 people, and we could see how they are likely to benefit from an anticoagulant by taking data from all over these places. Exploratory, it's not a trial, but now I'm making a model to say, oh, this type of phenotype or this profile is likely to benefit from an anticoagulant, or it's likely to benefit from a statin or whatever. And now then that informs which is the exclusion and inclusion criteria that I will try in my new trial. So it's one thing to use that way. We haven't done it yet, but this is some potential. Or efficient patient recruitment process. people are looking at registries, large databases in countries like Sweden and the UK that have a registry of everybody in the country, and everything that you do, they register. You go to the pharmacy and you buy a cough suppressant, that is registered. And if you go to any little hospital, or you go visit a medical doctor and it's private, the doctor comes to your home privately and he prescribes something to you, and then you go and get it at a pharmacy, that is registered. So now we know all the patient care, all of these things, and we could know who benefits, who's doing what, but who may be potential participants for my trial. Because now, oh, this person was getting something that is for irritable bowel syndrome, or is getting this. So maybe I can send them a letter of invitation. We are doing a trial, so would you please participate? There's ethical problems because those registries there's no consent to be contacted there. And so there are ethical issues in using this, but people are using these fast machines, search engines, to identify potential participants. This actually is not outside of clinical trials. This is what happens when we have our phones all the time. And I'm talking about, if I talk right now about, don't you like to wear blue color shirts? Your phone is going to start giving you ads for blue color shirts. Why? Because they've stolen that information and it's gone somewhere, and a search engine has identified, oh, they're talking about blue color shirts. Maybe they're interested in, now they sell that to somebody that sells blue color shirts. And now blue color shirts is something that you are going to be doing. So they're doing this. And there's no consent that you gave to your phone being listened to constantly. Even if you have it off, it's actually on. Sometimes you have to actually turn it physically off, not at dormant. So, but we are doing it for clinical trials in places that have large databases to identify potential participants, because it's a heck of a lot easier to recruit people once I know people are likely to be having this condition. We also want to get good participants. We want participants that will follow the protocol, that are likely to adhere well, that are likely to benefit. And we can do based on past experience from all other studies. If we have that information or access to the individual patient information from old studies, we knew who adhered. Oh, we know now that people between 35 to 39, males don't adhere so well as women that are this, unless that man is having diabetes, or unless that have this, or unless they have that. So all of these complex relationships that led to the adherence or compliance, or they missed 4 visits because of this. You can program it to say who is likely to adhere, who is likely to not adhere. And you can have these algorithms predicted. So now I would not recruit that age group. I will recruit from here. But, oh no, this is that. So you optimize who you're going to include in that. It's a great paper that recently came out that is there for recruitment and retention. Who is likely to complete the protocol? Who is likely to adhere to it? Yes.
Is it ethically correct to identify the subject who are likely to do better? And do you say it out in your method section? This is how we.
So yes, you have to. Well, whether it's ethical or not depends, right? If you are, for example, to search databases to identify potential people, it probably is not ethical. In some places, like for example, in Canada, it's not national, but in one of the provinces, in the province of Ontario, they have the Ontario Health Insurance program, and that collects the information of all transactions that are health related because they pay for it. And when you adhere to the health program, you sign consent. I can be approached. My information can be approached. So then it's ethical to use that database, but not everywhere it is. Some places it may not be ethical and people do it. But this is the ethics to approach you. They still have to get consent from you. They cannot randomize you or they cannot intervene on you until you go through this. This is just to identify. Now, whether you're a good participant or not, that's not a problem because now I'm using that information to optimize who I will approach. I don't think it's unethical to do it because this patient, I'm getting it from people that have already consented to being part of a study and providing their information there. And I'm for research. So nowadays we ask them, can we use your information for research? You consent because nowadays we do things that did not, 20 years ago, did not exist. So we can do GWAS on it. They gave their blood and we stored their blood, and we can do now genetic analysis to see if they would be, they have the particular SNP that has been identified by somebody else is likely to be somebody that will benefit from lowering blood pressure or having a anticoagulant or having this or having that. And so now we want to use that and we can use it. And then we still have to go to the person and say, we did this from your data that was you gave from part of the study, and that's why we're coming to you. Now, whether we do it or not is another thing, but then we should probably do that. But as she said, identifying patients, doesn't matter how you identify the patients, whether I do it this way, or I do it by Twitter, or I do it by this, I still have to get them to consent to join the study. This is just if it's ethic to approach somebody, right? The approaching is that different.
One is the generalizability. Also, you said to sacrifice the generalizability and the facilities where there is no such databases.
Right.
How do you say, for example, pool of patients have consented to participate in your study? Mm-hmm. Still, I can identify some of the potential patients who can do better in the study by taking their characteristics and modeling it and so on. So when I do this one, I am sacrificing my generalizability. It's not going to happen.
Completely agree. You are all you're doing by all of these optimizing who you include, you are optimizing that you will get a good result, a better result, right? Because these are people that are likely to benefit, but you're not, so you are affecting, as you say, the external validity or generalizability, but the study integrity is not affected because I'm going to randomize them to A or B. If I don't randomize, then we are in trouble, right? All clinical trials are in trouble if they don't randomize. But the internal validity is still okay as long as I randomize. Yeah, so that's, they're exactly true. Yeah, so people that are likely to be good participants. So these are things that can be done. Whether people are doing it is another one because of exactly these reasons. Now, one thing that's done is we talked about adaptive allocation, and one can use AI to optimize the adaptive allocation. Now, we haven't talked about, and I just sort of reserved it for this lecture, the classical adaptive allocation methods that have been done in the past. So we used to call them play the winner. So let's take, these are subjects that are being enrolled in my trial. The first subject comes in, and we have a 50-50 chance to go to A or B. So let's say they go to A. And this is for studies in which the outcome is known relatively quickly, not waiting until 5 years. But we have to know the outcome. Did A work for this person? Yes or no? If it worked, then the next person gets A. But this person, A did not work, so the next person gets B. And for this, B did not work, for the next person gets A. So you keep playing, the next person is going to get whichever was one before. And so it's going like that. This is for individual ones. You could also say there's another more complicated one is what is the number of times that A has been winning, number the proportion of times that B has been winning, and you modify it as you go along. People do this in some types of studies, usually in phase 2 trials. It's you're adapting who is getting what, right? People also do bias coin. They say the initial first person has 1-to-1, and so they get A. But I want to balance not the response. This is balance the numbers on them. So there's already one in A, but there's nobody in B. So the second person comes, I'll give probability 1/3 for A, 2/3 for B, because I want them to go to B. But I toss the coin and the 1/3 comes, the person gets the A. So now I have 2 people in A, nobody in B. So the next person, I change the odds, and you have now 1/4 chance of going to A, 3/4 chance to go to B. And let's say they go to B. So now I have 2 in A, 1 in B. So the next person, now I increase a little bit the chance to go to A. So I have now 2/5 and 3/5 here, and maybe A comes, and now it becomes 2/4 chance, etc. And you just keep going like that. So now what you're doing is that you're balancing out who is getting what, but it's not response driven. This is done so that you don't have to create your randomization scheme beforehand and hide it, because you haven't created it yet. You don't have to hide anything, right? And so you do it this way. This is also only done in small trials when they don't want to do the randomization scheme, and so not been done. But what I wanted to talk about, those were the classical ways we did it. If we wanted to do adaptive allocation. Now what AI is doing is covariate-driven adaptive allocation. So what do? I look at, I have a patient that comes in, 001. I know their phenotype. He's obese, he smokes, he does this, he does that. Da, da, da. I have their genetic, I have their blood samples, I have all the biomarkers on this person, all sorts of data. And from prediction models, I can calculate the probability that this person is going to do well on A and the probability that this person is going to do well on B from these models. And if the probability that it's going to do well on A is greater than the probability that it's going to do well on B, then I will assign him to A. But I cannot assign him because then I'm not randomizing. So I have to give higher odds for them to go to A than to go to B, flip the coin, that's a bias coin with the different odds, and then maybe they go to A or they go to B. But if their probability that they do better on A than B is less, then I'll give them lower odds for A for the next person based on this complex neural network prediction model. For each person, they run this model and see what it comes up. And then they start changing the odds for this. This way, they would optimize that you're assigning the person to the intervention that they're likely to better on. Okay, very complex design. I haven't seen it used, but there is this one paper on optimal allocation that's describing it. We don't do this, and I don't know who has done it, but maybe somebody has done it. These authors were this group, Liu in China, is doing. Yes. So you
about the outcome of, suppose it's a new drug, it's not being used and you don't have any covariates of how it will work, it won't work.
can't do this. So yeah, you'd have to have done previous study and base the models on that previous study and get those models from that, those neural network prediction models, apply it here, and you update it here. Once you know that that person do well on A or B, now you update it on this one, right? So you periodically keep updating your prediction model, etc. Right. So you may not get necessarily that they will be comparable at baseline from that. So there may be residual confounding. There's all sorts of complications on this. Oh yeah, for the response adaptive, you have to have the outcome recorded. So this is only done in studies that the outcome is known almost instantly, like surgical ones, or if you're looking at an outcome that is a drop in your fasting plasma glucose immediately, or something like that, or that your blood pressure changes immediately, or in the next 10 days, before the next patient shows up, right? So these are not applicable in most situations.
I guess it's much easier to randomize small batches of patients instead of one patient or the other.
Right.
Even if you have 100 patients randomized, here, 100 patients randomized, right? I can operationalize 5 patient batches, but randomizing one, having an outcome for one, and then waiting for the second one to appear, and then because if the second patient in this sequence, the second patient appears before the outcome of first patient, I won't be able to.
You have to wait, right? Yeah. Yeah. So yeah, you could try to modify the approach to batches, but then you have to wait for everybody in this batch to have it before the next batch is. So you still have the issue of waiting for the outcome. Yeah. I'm just telling you what they talk about, but I have not seen it done. What I have seen done is the following. We are the central statistical center for multi-center trials. Part of our role and responsibility is to monitor the progress of the study, that the study is being conducted well. We tend to do multi-site studies all over the world, and a particular study that's recruiting 5,000, 8,000 people will have 50 sites, 80 sites, 140 sites around the world. And you have to standardize the procedures, right? You have the common protocol. Everybody has to follow it. And if you have ever participated in a pharma study, you may be doing 10 patients, 5 patients. They pay you well to do those, but they send you this protocol that you must follow, right? And you get trained on it, or you may not get trained on it, but you get monitored by a site monitor that shows up one day without telling you they're showing up and wants to make sure that you're following the procedures perfectly. That is the way pharma tends to operate when they do the large scale studies. They want to make sure you're doing things like you're supposed to do, like they're paying you to do. So they check on you that way. We are not pharma. We are academic. We don't have the money to be sending people all over the world to check on things. So what do? We rely on the data comes to us and we check it. How do we check it? We use classical statistical models. What do statisticians study? Variances. Now, if, for example, and this goes back to nowadays, we measure blood pressure with devices that are digital and recorded nicely. in the dark ages of data collection, we had to write it on a piece of paper. And how did we take the blood pressure? With a sphygmomanometer. So somebody is going, pumping it, listening to the beats, and then writing down. Nobody writes down 120.4 over 87.3. 120 over 88 or whatever, or 120 over 90. There was always rounding. There was always a digit preference, fives or zeros. So they wanted it to be precise in this study. And they invented a machine that was a regular sphygmomanometer, but that the zero mark was random. And that was recorded so that they had to actually look at it, write down maybe 122 over 87, but it actually was a random thing there that was then collected and fine. They made this cumbersome machine. We also realized that blood pressure varies a lot. So they don't take only one measurement at the visit. When you come in, you sit for half an hour, they take 2 measurements. The visit lasts 2 or 3 hours. After another half an hour, they take 2 more measurements. After another half an hour, they take 2 more measurements. And at the end, they take 2 more measurements. 8 measurements every time a patient came in. They had to come in every 2 months for 7 years minimum. 4,000 people in 12 clinics. So about 1,000 or about 400 people per clinic. So using a cumbersome machine is very difficult. So one person, very smart person, decided, I don't have to do 8 measurements. I just take 1 measurement and I say +2, -3, +1, then, and I will generate the other 7, making up data, which is he thought was fine. And I'll randomly put +1, -2. We are never good random number generators. So if you calculate the mean of different, so we had the data from different people, different blood pressure takers, they all had to put in their code, and we analyzed not the mean by code, because the mean is going to depend on who you had, but the variability should be the same across anybody blood pressure taker. All of a sudden, there's one person that has a very small variance. This is suspect. So what did we do? We said to the PI of that clinic, just wander around and see what this person may be doing. That's when they discovered that this person was faking everything. What happened? The person, of course, was fired. His data thrown away. Luckily, this was not the primary outcome, because $120 million invested by the US government would have gone down the drain by one person faking data. In that study where they went through all of that effort to not have missing data, if you have fraudulent data on the primary outcome, forget it, it's gone. So this is the importance of making sure that you monitor things. So we used to do it classically in the old way by studying variability. At PHRI, we do central statistical monitoring to detect fraud, and we use a factor analysis-based risk score to identify fraudulent data. And it was constructed using basically factor analysis, principal component analysis, but it has to be developed for every study differently. And so more recently, we got a grant to use machine learning algorithms because they could identify with unsupervised learning anomalies in the data. We don't have to know what the anomaly is, because people that are creating fraudulent data try to be very clever. And we have to imagine, it's like a thief, right? A thief is going to be very clever. And so the police are always trying to be one step ahead of the crooks. And as soon as they figure out something, the crooks are already thought about another thing. So we don't know what patterns they're using for making up data or just being sloppy with data. So we had unsupervised machine learning for this irregularity detection by looking at patterns that were different, okay? And we use these things called support vector machines for detection of irregularities. Don't ask me what exactly they do. I don't do this. I had experts on machine learning that did this for me. And we got a pilot study published 2 years ago. And we just got the final paper where we took 8 different studies. We knew we had all the data from when the data came into us, before it was cleaned for a DSMB report, and after it was cleaned. So we knew what was cleaned. And can the machine detect it before it was cleaned? So we fed that into it. And so we were able to do form level irregularities and positive predictive value and sensitivity for it. And we got that we could do about 70%, 80% we could detect, which was a vast improvement. So now what we do is we target things. Instead of going to every site every few months and looking at everything, we now can tell the site visitor, go to these few sites, look at these few things, and look at that this way. So we target, we optimize the monitoring. And this is something that it's still computer intensive. We do it for some studies that have the budget to pay for the algorithms to be developed, et cetera. But the technology exists to do study monitoring using AI. I'm missing the I over there. There are other computer intensive techniques that are not machine learning ones, but this is to ensure balance across treatment groups. Before randomization, there's simulations that people do. Simulations take a long time of computer time to do it, but you simulate the different allocation process multiple times and assess the probability of achieving balance. So now you're trying to see, when can I get balance on that? You do it in multiple ways to do it. And say, okay, if I stratify this way, would I get balance? If I stratify that way. So you do it in past studies to inform how you'll do it in the next study. Or they do machine learning guided stratification is another one of these things. So they categorize individual into groups based on characteristics, and then they look at real world data like electronic health records, and they develop these risk assessment tools that will improve then their decision making in healthcare. So people are using this more and more. But just a concluding remark, that is that AI in clinical trials is in its relative infancy. It's another one of these sexy, attractive things that everybody, every day when I'm at my office, one investigator will come by and says, are you guys using AI for this? No, we haven't. Well, you should be. Yes, let's invest in doing this, but some things we want to do and some things we may not be able to do. But it's growing fast, so it's coming up. In fact, we already have concert reporting guidelines if you've used AI in your study. So this is now a new concert expansion for reporting. So if you've used AI in any aspect of your study, you need to then put that in your concert statements that when you submit it to the journal, for example. They're using it for optimal interventions, but I'll stop about that. This was that same, this is just a discussion in that paper on drug development of the different types of things that are the different types of AI methodologies that exist, and they use those for drug development ones. But I'll stop here with this one. Questions, comments? Totally.
But in the healthcare, I haven't seen that kind of robust infrastructure.
Yep.
So it needs a lot of investment.
Yeah, so at our university, there is a team that was hired by the hospital system and the university together. There are about 4 or 5 programmers, and they cost a lot. The machines are super fast computers. They cost an enormous amount of money. And so the university and the hospital system of the town have invested in this. And they're hoping that it will be useful. They're the ones that did this anomaly detection for us, but only after I got a grant from the National Institutes of Health of Canada to pay for them. And whatever money I got, 100% of that budget went for them to do this work. And they took them 2 years to do this work. And it was all the budget was gone for them. So it is costly, it is expensive. Now we have these papers were proof of concept. It can be done. But if we want to do it for a particular trial, we have to still pay them to do it. It's not. So the algorithms are reusable, but we had them customized for our particular case for this proof of concept. This next step to make them reusable will require more money. And I don't have that grant to do that right now. So we haven't done it.
Different features for each trial and the screening and the evaluation.
Right.
In every trial you have different things.
Right. And so this is part of the C disk and optimization of how data are collected, which data are collected, so that the features or variables are same. And so, yes, we would have to have something like that before it becomes readily usable. Now, we are just one institution doing one academic kind of research on it. I'm sure there are people in pharma in particular who are trying to do this to optimize the allocation, to optimize drug development, but it's not publicly available. It's intellectual property, and then they will be making the monies. And if Pfizer develops it, then Eli Lilly is going to compete, and Novartis is going to compete, and that's why it's so expensive for us in academia to have these 5 programmers, because they could go to pharma immediately and get 4 times their salary or 10 times their salary, because these people, these are computer scientists that are at the cutting edge of this machine-driven technology. But yes.
The fraudulent activity investigation, that once you found out there is a site which had this problem, did you find out whether the PI is involved or the technician involved?
Yeah, so this was the big question. Was the PI causing the trouble, or was it the technician causing the trouble? So the initial analysis, we had information. Luckily, we had collected information, and the CRF had every item that is filled. The paper had to say who completed this page, and there was a code.
Yeah.
So that's why we were able to do that analysis. Most people don't put that. It's filled in by somebody, but we don't know who filled it in, so we don't know who is doing it. In the other study, the study here that was that gave rise to this detecting fraud, the principal investigator of a trial was P.J. Devereaux, and his study coordinator noticed some anomalies in the data from just looking at it on a daily basis. She said that it looks like the people, these 10 people that came on this date, their data looks exactly the same as these other 10 people that came 2 months later. Is that possible? So she asked a human, now asks the investigator, is this happening? And it actually, this was not the data collector at that site. It was the PI at that site, because the PI at that site was being pressured to, you're not recruiting enough people. You need more people. We need more people. We're behind recruitment. You need to do this. You need to do this. So to get these people quiet, he made up more patients. It copy pasted. But this happened. So this was the PI that did it. So that all of the data of that site gone. And actually there were 3 sites that were doing it.
Do you involve them later on? Or?
So I would not. They were colleagues and friends of these folks, and I don't know what they're doing. I should ask PJ what he's doing with these people. So yeah, you know, you have, if you're doing studies where you're working, and we don't have a session on this. When you're working with multi-center studies or multi-site studies, if you're working with pharma, you are just a site. You don't know who else is working on this. You are not even actually a co-investigator on the study. You don't participate in the papers. You don't participate in changing the protocol. You are just collaborating by giving patients and getting well paid for it. You may be invited to a meeting, but you are not in the policy making. But if you are doing what I call multi-center study as opposed to multi-site study, you are a co-investigator. You and a group of 20 investigators around the world or around India decide you're going to study child development, and you have very few people. So now you all get together. Yeah, we all want to do this. You all together decide on the protocol that you're going to follow. You all agree that this is exactly how we're going to do it. You finally get funding to do it. Now you're doing the study. Somebody has to monitor. Who monitors them? You cannot monitor yourself, right? So you get a central statistical group to be the bad guys. That's usually us. So we as external statisticians, we are the statistical coordinating, and we say, you're not doing well, you're not doing well, and so forth. But if they are colleagues, they're friends, we cannot really be the police because we started off as friends and we end up as enemies. So this is not a good thing. So that's why the statistical group usually is separate from one of the investigators to keep things working well and personalities not causing trouble.
Sir, in the covariate driven analysis by AI, you have
It's
mentioned
not analysis,
okay,
it's
allocation
allocation.
by AI. The probability was determined based on the genetic makeup of the patient, right?
All sorts of things.
So will that not cause a sampling bias?
Sampling bias.
As more people get allocated to A and more less to B, will
No,
that
because
cause
it depends on who you're looking at, right? But you'll say, let's say we take all of these and know these people will do better with A, so they will have higher chance of being assigned to A. These people will do better on B, they have higher chance to it, but I will randomize still. I will not do 1-to-1 randomization. I'll do 1-to-1.62 or 1 to this or whatever. Yeah, the issue is just you may not get it to be balanced. Yeah, you probably will not get it to be balanced. So you have to do residual confounding secondary analysis. Would it be balanced?
Then you randomize.
No, but the randomization is with the preferential allocation
Yeah.
to the drug, which is likely to work right.
Right. So
With more risk factors, which will benefit from the drug.
So let's take an example. Suppose women are likely to do better in A than in B. And so we will be given higher odds for women to go to A than B. So at the end, we will have more women in A than we will have in B. So it's likely to be unbalanced on some of these characteristics. Yeah, that's it.
No, if the
Once it's becoming big, when the sample size increases, then it is a confounder, it may not be may.
No, it may be worse, right? Because the more people I have, the more we'll have in this.
My pretest probability, if I keep updating my pretest probability of allocation.
It just depends on whether it's a confounder or not, right? But it is, if they're likely to do, if women are likely to be doing better, then it is a confounder by definition. And so it's a known confounder. Yeah.
But initially you start giving, but if it is not really benefiting,
No. Then you're mixing up two designs. One is a AI-based allocation. The other is the probability. And each happens in
Separate.
Yeah, am I right?
Yeah.
If you go to AI-based allocation, then it won't give the prior information on whether the drug works on the patient.
Yeah, it's not, it's covariate driven. It's not response driven. So these are three separate things. They're not, you don't put them together. But yes, you're right. If it's covariate driven, the allocation, then by definition, the distribution of these covariates is likely to be different between A and B.
I would like to.
Either A or B, on what basis?
No, not like that. No, not like that. Let me explain that. The question has a lot of dropouts, for example. There are people who do not want to come back again and so on. So I wanted to use AI who is more likely to be participating in future. Good people to get involved in the study. That's one part of it. So use AI to select those people. You have 100, finally you have selected 20 out of using AI to get involved in the study. Then these 20 people will be randomized to go to A or B.
And that's okay.
That is okay. It still raises the generalizability. Okay. So you have taken only good people. So the other one is covariate driven allocation. That is whether women will do better, men will do better. Different weightage given to the subjects. That is different.
And that one is done
Yeah.
basically to help you in recruitment. So now I've selected these people that are likely to do good. They still have to consent, right? And they may not consent if they feel that B is not greater than A, or they don't want to be randomized to what they think may be by bad luck, go to the one that they think may not be good. They don't understand equipoise. And you say, no, we're going to give you, we know your profile. We know that you're a non-smoker, a diabetic, non-diabetic, that you are female, that you're this or that, etc. And so based on that, we will give you a probability of being randomized to the thing that's likely to work best for you. That's what they said.
Also, so ethically, right?
Yeah, so it's ethically, I'm going to give you what I can predict based on my prior information is likely to be the best thing for you.
So by comparing this layer and AI covariate driven, in the first case, the second participant will be recruited only after the outcome of the first has been completed.
No. In these two scenarios, the second one is only after this one is completed. In this one, they may still be doing something. It's just how many people are in A versus how many people are in B. They still may be going on. Here, it doesn't matter. It's just this next person comes, I'm going to change them the odds, and then they continue working on whatever for 2 years, 5 years, 10 years. And now the next person is recruited, I will assign them with a different probability, and they continue and like that. So the only one that depends on response has to wait till the response. These don't depend on the response. So you don't have to wait on, response
That's mainly on the basis of probability.
for the baseline probability. Yeah. No, it's just your generalizability, as was said, that our study is not everybody that has this condition that meets this eligibility criteria. Our study is everybody that has this condition that meets this eligibility criteria and is a likely person to be a good participant. That's your true target population.
If I select the more population that are likely to complete the treatment,
Mhm.
and suppose one here is not using the AI-driven selection.
No. Everybody has to do the same thing. In a multi-center study, everybody does the same protocol.
AI is always there.
No. If you're doing a multi-center study, there's a common protocol. Everybody has to follow the same one. We had one case in the United States when they do trials, multi-center trials, they do a proposal or a grant to a coordinating center. They do grants to the different institutions that want to be sites. Well, in this one particular study, 24 sites were selected. And so then they married together those 24 with the coordinating center. So all of us now are working on the same study. We have to get the same protocol out because everybody's going to follow the exact same protocol. Well, there was one site, the investigator at those meetings said, no, I don't like that way you're proposing to do it. We're not going to do this way. And they all tried to convince him, no, this is the way to do it. Meetings were taking long time, discussions were going on. At the end of the day, this guy said, fine, put that down on the protocol, but I will not do it at my site. That was in the minutes of those meetings. The NIH took away the funds for that site. Drop. If you don't follow the protocol, you're not part of the study. That's why you have a common protocol. All right, lunchtime. Oh.
AI is in analyzing the data. So all how AI in analyzing, because most of the time it's easy for me to give ChatGPT one publication, one research, and ask them, this is my hypothesis, this my study question is this. Please give me sample size, just for knowing how much is the samples I have to collect.
Okay, so it depends what you use the AI for. If you used it for sample size calculation, if it's the correct one, sample size calculation, who knows if that ChatGPT gave you the right calculation, or if it took into account the potential loss to follow-up, the potential crossovers. Did it take into account the lag time in recruitment? Did it take a lag time into so crossovers, loss to follow-up, and the recruitment period, and so forth. So some of these may or may not, I don't know. I've never used ChatGPT for sample size calculation. But if you use ChatGPT or you use AI for determining who to include, that doesn't matter. It's just, or how to recruit. As long as you then randomize, doesn't matter. Now, if you're doing this covariate driven allocation, you better deal with residual confounding, because that's, so now it's going to impact potentially your intention to treat, because your intention to treat dictates that your primary analysis is unadjusted. And so you may get a bias if you do this. And you don't, and that fight the first one. But then once you do the adjustments, you may get a result. People will not do this if you're going to get a big bias. So why you would not use covariates that are likely to be confounders. You would maybe do it based on who knows what. But then, so there are problems with this because it affects your analysis if you use some of these things. But if it's who you recruit, and what else? You could do it for anomaly detection, for monitoring, that doesn't affect your analysis. It tries to improve your quality of your data. So depends on what you use AI for in these things.
So, sir, we have not started using AI for analyzing. Like it will make easy.
What would it
Will
make
decrease
easy?
the
Is the
time.
analysis is easy already? The analysis is a t-test. What else is easier than that? Or a log rank test? You're talking about secondary analysis where you're trying to torture the data to see if it confesses to somebody else. But then you will do all sorts of models, you will do all sorts of things. That's separate. That's not your primary analysis of the trial, right? Yeah. You will not take away our job. All right, we'll discuss more after lunch.